Good morning, everyone. Welcome to Wednesday of NACCB 2022. I hope you're continuing to enjoy the conference. I'm Jessica Pratt. I'm the outgoing Vice President of Education and Chapters. And today it is my great honor to present our 2022 Student Awards for Best Posters, Speed Presentations, and Full-Length Presentations. So I'm hoping that people are coming in from the lobby and that many of our award winners are going to be here with us this morning. I will just dive right in. So if your name is called as one of the award winners, you can make your way up here, and Becky has plaques to hand out. Our Best Poster Presentation Award goes to Jeremy Adkins. All right, Jeremy, are you here? We'll check in a minute. Jeremy's poster was titled Pinus monophylla, Seedling Trait Variation and Performance Under Various Seasonal Precipitation Regimes in a Field Common Garden. We have 2 awards for speed presentations. Our Best Speed Presentation Award goes to Greta Schmidt. Okay, maybe Greta's not here either. Her speed presentation was titled Multi-Scale Responses to Urbanization by a Southern California Felid. The Best Speed Presentation Award with Conservation Application goes to Leah Chalifor. I may have to walk through the lobby with my list of names and see if we can round up some of these individuals. Leah's presentation was titled Identifying a Pathway Toward Recovery for Depleted Wild Pacific Salmon Populations in a Large Watershed Under Multiple Stressors. Now for our full-length presentation awards. Our Best Full-Length Presentation Award goes to Melissa Cronin. Congratulations, Mel. Melissa's talk was titled Estimating Shark and Ray Catch in Tuna Fisheries to Inform Assessment and Conservation. Our final award for today is Best Full-Length Presentation with Conservation Application, and that award goes to Christopher Jadala. Congratulations, Christopher. Christopher's talk was titled A Means and an End, Harnessing Community Science Literacy for River Restoration. So if Jeremy Adkins or Greta Schmidt or Leah Chalifor are here, or any of you know them personally, you could send them a quick note that they should come and find us after the plenary session this morning to receive their awards. We're going to have a quick photo op here. All right. Congratulations. And now I'd like to pass this over to Mary Peacock from the University of Nevada, Reno, who is going to introduce herself and our plenary today.
Hope you can all see me. I am very short. All right. Hello, all. It's my great pleasure to introduce Lisa Heki today. She is the project leader for the U.S. Fish and Wildlife Service Lahontan National Fish Hatchery Complex. My name is Mary Peacock. I'm a professor at UNR and a conservation geneticist with a large research program on Lahontan cutthroat trout that is in no small way a result of Lisa's vision and support for this species. It was my suggestion that she be our plenary speaker because I could think of no one better to tell the conservation story of Nevada's iconic cutthroat trout. Lisa and I have been working together in the trenches of LCT recovery since the 1990s. And believe me when I say this, we've seen it all. In fact, we almost got into a fist fight in a parking lot with a guy determined to undermine cutthroat trout recovery. Ah, good times. But I have to say, and I do not exaggerate, Lisa has single-handedly resurrected the iconic lake form of Lahontan cutthroat trout, now called the Pilot Peak strain, that is being raised at the hatchery for conservation and restoration activities. Lisa and I, a manager and an academic, have been true partners and colleagues in Lahontan cutthroat trout recovery and working through its challenges, which continue today. I have to say that working with Lisa and her team has been a true collaborative and rewarding experience. She has vision and she's tenacious, ask anybody who knows her, but I will let her tell that story.
I'll have to move the microphone up a little bit. Mary and I are polar opposites in the height department. So, thank you, Mary, for those generous words. And it has been a nearly 30-year collaboration on this story of Lahontan cutthroat trout. And I've learned a lot from you, and we've made some substantial progress in the community and understanding that, yes, the strain that we're using is related to the original Tahoe pyramid. I'm just going to jump in to talk to you a little bit about the outline and the arc of my presentation. So it's going to span my station, my current contemporary configuration of my program and what we focus on. I'm going to touch a little bit on Pleistocene Lake Lahontan, which is an inland lake that formed in the Pleistocene. Briefly touch on the LCT biology history in case you missed some of the sessions that covered some of Lahontan cutthroat trout recovery that Mary held, and some great speakers presented on far more detail than I'm going to go into today. I'm also going to touch on the most significant impact to the Pyramid Truckee watershed in the history of this species and Pyramid Lake ecosystem. I'm going to also touch on some conservation activities that went on before I came on scene, and go through briefly the rediscovery of the Pilot Peak strain of Lahontan cutthroat trout, the stocking efforts that we have pursued in partnership with the Pyramid Lake Paiute tribe, the monitoring and research activities that we're working on in collaboration with universities and USGS. We work together with science research so that we can publish our progress. So, my program is currently configured in a way that it wasn't when I started in '92. It has expanded, shall we say, over the last nearly 30 years. I currently have Lahontan National Fish Hatchery, which is an hour south in Gardnerville, an hour south from Reno. I have a Marble Bluff Fish Passage and Research Station, which is an hour east along I-80 in the Truckee River corridor, and it sits at the terminus of the Truckee River near Pyramid Lake, and I will go into the origin of that structure and facility. And most recently, in the last 15 years, Fish and Wildlife Conservation Office, that the majority of my 23 staff reside in Reno. And all the folks work on all of the programs: hatchery, fish passage, monitoring in the Truckee, collaboration with researchers, and also at the hatchery, so that everyone can be aware of our program priorities, how we function as a team, and have a little empathy for how different tasks require understanding, patience, and keeps everyone on the same page. So this is an unusual program nationally for the U.S. Fish and Wildlife Service. So Fish and Wildlife Service has 3 programs: refuges, they have ecological services, which is a regulatory branch of the Fish and Wildlife Service, and then there's fisheries, or fish and aquatic conservation is what it's currently called. And my shop is within fisheries. But as I mentioned, we are an integrated program doing a variety of work to focus on a holistic approach to conservation for both Cui-ui and Lahontan cutthroat trout. So watershed restoration connectivity, not just in Pyramid Truckee system, but statewide with our National Fish Passage program. We manage Truckee River in-stream flow for the benefit of listed fishes of Pyramid Lake. We are also involved, close partnership with the Pyramid Lake Paiute tribe. None of this would be possible without them. We propagate Lahontan cutthroat trout at Lahontan National Fish Hatchery, and this is a unique strain that is carefully propagated to meet reintroduction as well as recovery efforts. We also, as I mentioned, we fund and collaborate with a variety of researchers. I have done that throughout my career, knowing the limitations of our, we only have so much capacity. I'm not a researcher, I'm not a geneticist, I'm not a geomorphologist. And the growth of myself personally, the program has resulted from working with pragmatic researchers, understanding the real world challenges that we face in conservation, and kind of melding those two together that helps us move forward productively and moving the needle, an overused phrase, but moving the needle. And again, ongoing partnership with Pyramid Lake. The Fish and Wildlife Service has been in partnership with fishery management and Pyramid since the 70s, before my time, and we couldn't do it without them. And this is a picture of my crew. It was a picture I took when we first got the first ping on a pilot peak migrating up the Truckee River from Pyramid Lake, and we were pretty excited. I mean, the commitment, the dedication. I have some folks from my shop here of just the remarkable moment in time that we all got to observe and what they contributed to this moment in time. So really quickly, I'm going to go through some of these initial slides pretty quickly, but basically Lahontan cutthroat trout and the native fishes in Pyramid Lake all evolved in this ancient Pleistocene Lake Lahontan, and look at it as an inland sea, high alkaline, high saline, and it receded with the end of the last ice age, and the decline left remnant population of habitat for Pyramid, Walker, Honey Lake, and Winnemucca Lake. And really a unique, rich ecosystem that contributed to the age of these species to each other. Cui-ui, Lahontan cutthroat trout, large size, long-lived, just a really unique system, and they're all still intact in Pyramid. Can't say the same thing about Walker or Winnemucca. So we're going to focus, going forward fairly rapidly, the Truckee Tahoe Pyramid watershed, and that is where my shop, our partners, focus on reintroduction and recovery of the native strain of Lahontan cutthroat trout. And if you notice, if you can see Lake Tahoe in the Sierras, and it flows outward only east into the Truckee River, and that Truckee River terminates into Pyramid Lake. And terminal lakes, they have no outlet, so think of it as an inland sea at Pyramid, and so the cutthroat trout, the Cui-ui, all migrating from the quote-unquote inland ocean upstream to Truckee to spawn, reproduce. So some of the basic biology, they're an inland cutthroat trout, one of 14 subspecies, the only trout native to the basin. A lot of non-native game fish have been stocked through the last century, but Lahontan cutthroat trout are the top predator. There are two life histories. We focus on the lacustrine form or the lake form and reestablishing them in their historic habitats. They are the top predator. They grew to a very large size, both in Lake Tahoe and Pyramid, and were just unique fishery, particularly the turn of the last century, the overharvest, some of the habitat degradation in the Curve and Tahoe Pyramid really contributed to the challenges. Which one are we looking at? So, just covered those. So, Lahontan cutthroat trout, culturally significant to the Pyramid Lake Paiute tribe, culturally significant to the Washoe tribe of California and Nevada in Lake Tahoe, and they provide also an extensive recreational and commercial fisheries. Then they were extirpated from both basins in the 1940s, as well as Walker Lake, as well as Winnemucca Lake. Well, Winnemucca Lake, they were extirpated by the turn of the last century, but I'll get into that sad story. So, extensive commercial harvest. They used to put these big trout. When John C. Fremont came across in the mid-1800s, he quote-unquote discovered Pyramid Lake. That's who named Pyramid, based on a structure out in the lake. And he called them the salmon trout, partly because of their large size, but also the flavor. They're very unique in size. And this is a Lake Tahoe fish. The individual you see standing there, that is a Lake Tahoe fish. Though it's more oligotrophic in Lake Tahoe, they did reach a large size. And, you know, later in the last century, with the decline of LCT due to logging, sawdust choked the Truckee River outflow. And then Derby Dam was completed on the lower Truckee River in 1905. So Derby Dam, and it is on the Truckee River again, about 38 miles upstream from Pyramid Lake. And it was constructed, the first Bureau of Reclamation project in the Truckee, in the nation. Klamath Basin was close behind, but it was an era of wanting to make the desert bloom in a parallel basin in the Carson River Basin. And the structure was put cross-channel across the main stem Truckee River, again, just about 40 miles upstream from Pyramid Lake. There was no fish passage. They attempted some fish passage in 1918 and maybe a couple attempts after that, because some in the community knew that we were losing this unique migration of LCT. And the powers that be, similar to salmon, Columbia Basin in the 50s, you know, building dams, there was a conscious knowledge that those would disappear. The salmon would struggle in that fragmented habitat. So they historically, from 1905, took all of the river most of the time before 1967. And with the terminal lake, this creates significant changes to the river process, river function below Derby Dam. So not only could Lahontan cutthroat trout not reach their upstream habitat where they would have spawned and incubated their future year classes, the habitat became more and more degraded in a rapid way. So Winnemucca existed prior to the construction of Derby Dam, 1905, and it was an overflow from Pyramid Lake, and it was made a refuge right before the lake, about 1895. Winnemucca was made a wildlife refuge, but it had Cui-ui, it had Lahontan cutthroat trout, it was a huge migratory bird path, and then Derby Dam was built. And you can see the rapid decline. The Bureau, the Newlands Project operators would shift the entire Truckee River during peak runoff, when Cui-ui are in the river, when Lahontan cutthroat trout are in the river, and pull it entirely over into the canal to take it over to another river basin, and leaving thousands upon thousands of fish stranded in the lower river in a dry riverbed. So the decline in elevation, 80 feet to 1967, was rapid and destructive. So this is a picture in 1977 without in-stream flow, without the variability of in-stream flow. The cottonwood recruitment ended below Derby Dam. The channel was braided, shallow, hot. The river looked slightly better. When I got here, there was 1 year class of trees produced in 1987, but the river looked pretty much like this when I got here in 1992. This erosive force of a declining terminal lake and a river that's trying to degrade to meet that new elevation of a pyramid of a lake system moved a lot of sediment downriver. And as you can see, this again is in the 70s. This huge delta formed, shallow, spread out, fingers of flow from the Truckee into Pyramid Lake. This hampered fish passage, caused stranding, continued erosion, and then predation by pelicans. There is a pelican rookery just offshore where one of the largest pelican rookeries, and so pelicans hang out. They're trying to reproduce. They've historically used it as nesting grounds and utilize the fishes of Pyramid Lake for reproduction. So this erosive head cut, you can see this is a relatively newer picture. It's probably early 90s, but this is Marble Bluff Dam. And it is a structure that was built in '75 as part of the Washoe Project Act, the Bureau trying to mitigate for the impacts of the Newlands Project as well as upstream reservoirs. And it stopped the head cut. But then it had created a 40-foot gradient shift from below the dam to above the dam. But it did stop the head cut. There was a 3-mile fishway built to parallel the Truckee below Marble Bluff because of the braiding, the delta that had formed. Fish couldn't get past it. And it had its challenges when it was constructed in '75. There was a fish passage facility in there. Everything was built designed for salmon. No consideration for Cui-ui or Lahontan cutthroat trout. So these impacts from 1905, the devastation that occurred to Pyramid Lake, to the lower Truckee River functionality, habitat, passage barriers, and then the Endangered Species Act came online in '67. Cui-ui was one of the first listed as endangered in 1967. Lahontan cutthroat trout listed as endangered in 1975, range-wide. So as a unique opportunity arose in the late '70s by the Pyramid Lake Paiute tribe. When Stampede Reservoir was built, they put in their purposes, it was supposed to be an M&I reservoir, they put in their purposes that it was also could be used for the listed fishes of Pyramid Lake. So you have to keep in mind the Endangered Species Act was relatively new, and no one knew what the unintended consequences of being generous would be. And so the tribe sued the Department of Interior, and one, Judge Solomon, sided in favor of the Pyramid Lake Paiute tribe, so that purpose of Stampede Reservoir, which is a significant moment in the conservation of this watershed, went right to the top of the list for its purposes. And it has was used exclusively for Cui-ui migration and spawning from '82 through 1994. I'll get into why it was used just for that species in a moment. So the 1990s. So here are a couple of paradigm shifts, opportunities that came about at that time. So ecosystem flows became an emerging science. So I talked about Cui-ui, and Cui-ui are only in the river for about 6 weeks. Their fry emerge within a couple of weeks, depending on temperature, depending on flow, and they all get back out to the lake, which makes it a nice, tidy water management modeling exercise. And the recovery plan for Cui-ui was based on IFIM. And you need a single species, a single life stage, and you use the lower Truckee River basically as a spawning channel. You throw water down in that degraded system to maintain temperatures. And then the river was dry for the rest of the year. For 6 months out of the year, the lower Truckee River was completely dry. When I got here in '92, it was completely dry most of the time. So microsatellite markers, a conservation genetics leap forward in being able to manage populations and their associations. Bring in Mary. There were a couple of other geneticists. A little side note, I did talk to in 1994 Shiwazawa out of Utah State University because he was extracting microsatellite DNA out of human mummies. And I thought we could confirm the origins of some of these out-of-basin populations that might show that they were the original population. I only had 5,000, and it didn't work out because we didn't have the museum specimens. And we didn't get those until Mary came on, about 8 years into the program, about 8 years later. But I thought I'd give it a shot. And these ecosystem flows, while I was not aware of them at the time, I did hire and work with a geomorphologist who knew a cottonwood researcher in Lethbridge, Canada, and we collaborated work on how to manage flows for the ecosystem, for the Truckee River, for river function, for forest recruitment and maintenance through time, thinking that building the blocks for both Cui-ui and Lahontan cutthroat trout was a focus. So public law was another momentous phase in the history of this conservation effort. Senator Reed authored this legislation and emphasis on water settlement. A lot of disputes, Samuel Clemens, whiskeys for drinking, waters for fighting over in the West. And it was a bruising battle for most of the last century. The Ore-Ditch Decree was finalized in '37, which managed the water rights in the basin, specifically for agriculture in Nevada. And it was the legislation that the funding that hired me. I was responsible for Section 207A and B of the Act, expedited implementation of recovery plans for Lahontan cutthroat trout and Cui-ui, restoring the Truckee River, and improving fish passage. So that was my job. I was hired by the Hatchery, and that was my focus. And I guess I took it seriously, and I pressed forward and wanted to develop a new paradigm for LCT and Cui-ui recovery and the Truckee River, and not just use the Stampede Reservoir water storage for one species for a short period of time while the river is dying annually. Looking back, I was 32 when I started, looking back, the water settlement piece, Senator Reed, the powers that be, wanted to expedite getting recovery plans done. The 95 plan for Lahontan cutthroat trout was completed in 1995. It was the first recovery plan for that species, and it had been listed as endangered in 1970. It was downlisted to threatened with a 4D rule in 1975, and they wanted to get recovery plans written so that they could finalize the negotiations for the Truckee River operating agreement. You know, I didn't understand that as an incoming biologist responsible for recovering two species and an ecosystem, not just one species. But they developed a model in the Cui-ui recovery plan, a nice tidy model that showed we need 180,000 acre feet for Cui-ui, and we're just gonna throw it down there at the time. I shouldn't say that. That didn't sound very graceful. We're going to release it during that window when Cui-ui need it, and then we'll turn off the tap and let the river be dry and the trees continue to die. And once you lose a cottonwood forest, they're gone forever in the Southwest. And that's what I learned from the researcher from Lethbridge, Canada. So anyway, there was a huge impetus to get recovery plans written. The LCT recovery plan did not include the lake forms in Tahoe, Truckee Pyramid, despite the rumors and the information that there were these out-of-basin populations, one in particular in Utah, that were believed to be original Pyramid Lake strain. And I found that challenging for my conservation directive. I needed to expedite recovery for both species and the habitat. And so I pursued avenues independently. I was the only inland trout program in the region, Oregon, Idaho, Washington, California. And so, we weren't a focus in these regions. And so I was able to make some headway. And also Senator Reid supported some of our vision for recovery and saving Pyramid and Walker Lakes. I won't get into Walker Lake, but anyway. So with the partnership with the researcher from Lethbridge, Canada, the geomorphologist and myself, I was given operation of Stampede in '95, so we decided to take a look at using the water in Stampede Reservoir to try to reestablish the variability in the hydrograph that was there historically, not necessarily the volume, but the variability that these western waters on the eastern Sierras require for function, for health, and to use that as a watershed scale river restoration vision, wherein you could get natural reproduction of willow and cottonwoods. The channel would narrow, temperatures would lessen, would lower with a healthy, deeper channel, and you wouldn't need to use as much water in any given year because the channel is healthier, the temperatures are better under lower flow conditions. And so we practiced it, 95 to 2000, high water years, had a great time trying to keep the decline of the hydrograph from the peak melt from dropping faster than an inch per day in that era. We're doing it an inch every 3 days now, but and the trees, if you're driving down, I know we did a field trip on Sunday, you're driving down and you see all those cottonwood and willow trees, that is a managed flow regime that mimics the variability that that river once saw and that was reestablished. And so that is a huge building block that began early on in my career, and droughts, high floods are all good for riparian forests. Not extended droughts like the one we're in now. But so once I could demonstrate through the establishment that we could use Stampede Reservoir water in a different way, and made the switch to creating a variable in-stream flow, not just for habitat, but for the listed fishes in drought years. So these are flows we designed that mimic the hydrograph. If it's drought year, earlier melt. If it's a wet year, later melt. And May migrations for both species. Well, LCT wasn't in the system at the time. So anyway, so that's a huge building block that was happening in the mid-90s. And then moving to Pilot Peak. So because they said we didn't need to recover LCT and Pyramid and Tahoe because the population was extirpated originally and that it wasn't a priority to focus on in the recovery plan, I got a line, I had a 3-page comment letter, and I got the line, you can investigate the ecologic and genetic importance of the lake form. That was what I got in the recovery plan. Proceeded to work with Utah State, game manager, Bryce Nielsen, who had been working with a private landowner on this stream, Morrison Creek, in the Pilot Mountain over in Wendover, Utah, an unlikely place to discover such an amazing genetic legacy. And Robert Behnke, only having allozyme and mitochondrial, I'm off my, it's not my, but that's what they had at the time. And people said, we don't need to recover it because it's gone, and the habitat's gone, and it will never be the same, and it's not recoverable. And I found that I didn't like that answer. And I proceeded on, and I can still remember the face of the person that wrote the LCT recovery plan when we flew fertilized eggs back from the ponds in Utah to Lahontan National Fish Hatchery. Let's just say I wasn't popular that week either. And we started a long-term frustrating process of building a brood stock from that original strain. Well, at the same time at the hatchery, we were also a put-and-take hatchery, which, hatcheries have their mixed history and is not my background, but they can be a useful tool in conservation if done well and conscientiously and not for mass production. There can be a use for them. And that's what I wanted Lahontan National Fish Hatchery to be. But I was balancing an existing put and take with a population that we were working with the tribe to maintain a fishery out there. And anyway, so we had a garage, we retrofitted a garage. Anyway, it's grown up since then. But, and we no longer raise the other strain of LCT. I turned that program off in 2002, and we were only trying to raise pilots at that point. But you can see the size of the creek. But the private landowner built ponds with the Utah Fish and Game, Bryce Nielsen, and they lived a long time. There was a 15-year-old fish out there in the ponds, 10 pounds, so you knew they weren't the same. They're not a stream fish. And so anyway, we had a great partnership with Bryce Nielsen. He was very excited that someone wanted to make use or investigate this strain of LCT. So I'm building the watershed health, connectivity, riparian forests, building that home, looking at what is the original strain and trying to, convince people that it was still on the landscape and that it could still be a valuable tool in recovery and bringing that fish home to its waters in Pyramid in Lake Tahoe was my goal. So broodstock management is complicated and challenging. We started at 95. I had to erase the entire population in 2010. We started over. Mary began to work in collaboration with us to be more effective in our pairing and matching to maximize genetic diversity, to not have any hatchery influences on this broodstock. I wanted the broodstock genetics to match the Wendover pilot peak in Utah, to match the production fish so that they were all one and the same. And Mary has worked with us annually to set up spawning matrix, to optimize the pairings. We installed a chiller so that we could match the temperature fluctuations in Pyramid Lake in the fall and what the temperatures would look like, and we used historic data in the Truckee River, and the chiller mimics that actual data for the broodstock. The broodstock life is kind of a sad existence. We do not stock them out. Somebody asked me that on the field trip, and we tried early on because I don't, it's a tough life, but they just don't do well in the wild, and it gives a bad impression for Pilot Peak LCT. But the chiller has had major influence in this temperature profile, major influence. February, they become ripe, and we see this peak that is matching what we see in the wild. I'm pointing to my crew to correct me, but the peak that is happening in the wild. And we keep future brood in these little aquatic habitats. There's like 10 crosses in each of these little blue habitats. It's highly intensive, labor intensive, and the little bags are a cross of one optimized male with one optimized female. Oops, the crew labels each bag. We track each bag through its lifespan. And that's Corrine, our broodstock coordinator, who has worked most closely with Corrine on the matrices and the spawning. So we sort on Monday, we spawn on Wednesday, and we track it through for production and future brood. And Corrine has moved on, unfortunately, and so we're working on that. So with this propagation program, we've been able to put 2.25 million pilot peak stocked in 2006. There was a delay in getting fish into Pyramid. There was a lot of stumbling and missteps, and the Pyramid Lake Paiute tribe had worked on their fishery since the 70s. They led that program once NDOW was no longer the responsible entity for Pyramid Lake, and it required a a level of time and dialogue and education with Mary, and that's some of the discussions that we have had through the years, to see the benefit that now we can call this population a recovery population and not just a put and take fishery. But it did take some time to get us there. So we began stocking in '06 and have just been amazed at the results. So the Pilot Peak strain, the genetic legacy assumed to be long-lived. Cui-ui lived to 50 years, reproductive to 50 years. I'm assuming LCT, the Pilot Peak strain, will live, I've already put my number on it, 20 to 25 years. You know, obviously fewer of them at that age, but nonetheless, I see long-lived along with Cui-ui, along with the Tui chub. We've been working in partnership with the Nevada Department of Wildlife to stock Pilot Peak in the Truckee River as part of a put-and-take fishery. I'm not trying to establish a river population, but to keep them from putting rainbows in, because that is still an ongoing challenge. All of our Pilot Peak are adipose clipped. So the remarkable thing, we got them in '06, and I assumed that they would be late at reaching sexual maturity based on the articles describing the original population. And I would tell, given that everything on the Truckee was water-based, water rights, water lawsuits, that I tried to convince a regional director that, you know, cutthroats would only use the river in big water years. And they showed up in the driest of dry in-stream flow, which is good. They're very plastic, they're very adaptive, they're very resilient. I know that's the theme of the conference, and if you want an image or a mascot for resiliency, Lahontan cutthroat trout and Cui-ui. And Cui-ui are just so cute. And they look ancient and wise beyond years. They're about this big. Anyway, so a tribal member, I think an elder actually, looked down over below Marble Bluff Dam. So Pyramid Lake should be at the top of Marble Bluff Dam, but for Derby. And so the lower river below Marble Bluff Dam is lake bottom, and it's not my goal to have LCT spawning there. We need them upriver, up to the tributaries, up to the Sierras where they originally were. And they showed up in a drought year. And there went one of my theories out the window, that they show up in all kinds of flow. And it's really excellent. So my crew got together. As I said, our first order of business is to document reproduction for the first time in 80 years. And so they did. And you can see the little vial there with Pilot Peak LCT reproduced for the first time in 80 years. And once they got into their home waters, all of those life history characteristics that we had hoped in many genetic presentations, trying to convince people that we still had this population on the planet and that it could thrive in its home waters, was now a reality. They were in people's hands. Fishermen could fish for them, and it wasn't just theoretical anymore, it was tangible. And there's Tom. So since 2014, the crew has been masterful in adapting and modifying both Marble Bluff, because the fish keep getting bigger, and so the crew has to keep extending, a little bit braggadocious, have to extend the measuring tables because the fish are 25, 29. We're hoping for a 30-pounder soon. But as you can see, the blue line is Pilot Peak LCT, and they just keep getting bigger and more abundant in the migration in the Truckee River. And it's just every year is an exciting new opportunity to understand how this fish once used the system. And it is a modified system, and we all understand that. We don't want them reproducing below Derby Dam as the optimum recovery, but it has been, for me personally, just a thrilling phase in what has been a struggle to move the system from what they wanted to what it needed to be, multi-species ecosystem function. But here we are, and I have just an excellent crew and so many moving parts, and they lead it, they manage it, they brainstorm it, they work with researchers on the ground. And so we have, some of you went out to Marble Bluff Fish Passage and Research Facility. That is the official name of the Passage facility. And some of you may, coming from, UC Davis labs or other fancy labs, Marble Bluff is a little more rowdy than that kind of a research station. But we get a lot of research done. We do adult LCT monitoring in the lake and in the river. Juvenile LCT monitoring in recent years with Robert Alchokachi. We are annually monitoring juvenile reproduction, waiting for that first F1 to call it a naturally reproducing population. We're close. If we haven't already, we haven't run those genetics, but we're pretty close, right, Roger? So again, here's a Marble Bluff, built in '75, stopped the erosion, and it is now the focal point, because now we have a natural reproducing population. It is now the focal point of our complex. The hatchery is key in that it continues to provide production for our Tahoe reintroduction programs, for recreational opportunities in the Truckee River, for education purposes, so that we can bring a variety of communities along with us, because they can fish for this species, 4D rule. But now this is our focal point, managing a wild population. So again, I didn't realize we had been funding Utah State since 2011. I've had Terminal Lakes funding for many years, another supporting program funded by some of Senator Reed's appropriation in his attempts to save these terminal lakes. And Phaedra's lab has been out there working with us, and we with her and her folks and her graduate students, because we want to understand how LCT pilots in the system use the system, what's their prey base, what's their growth rates, what's their survival, what's their performance, and make sure whatever we are stocking in Pyramid Lake, not counting any natural reproduction that will occur into the future, but make sure the carrying capacity, that we're mindful of how we're stocking this habitat. And then, again, working with Robert Alchokachi to monitor reproduction in the Truckee River from the time they show up in, depending on the water year, February through May. The first red we saw in 2014 was just something I had never seen in my career. There have been a population of LCT, as I mentioned, a contemporary strain that the tribe had managed and we had managed in collaboration with them, but it was not native to Pyramid Lake, Summit, some Carson River genetics, et cetera. And they never, 200 fish a year throughout my career, and that's still, give or take, still the same trend, although they're trying to catch on because pilots are now in the system, and I think it's helping them figure it out. But anyway, no signs of natural reproduction. And these pilots showed up, became sexually mature, and these reds are, 10 feet long, 4 feet deep. They're salmon, they look like salmon reds, and I have never seen anything like it. And just incredible. And that's in, some pretty marginal habitat for reproduction, basically lake bottom. And the crew has built some methods to monitor fry, timing of emigration of fry, that coincides with our water management strategies for wet years, normal years, dry years, because we want to track how they're using the system in variable hydrologic water conditions, how quickly they leave the system. So it looks like Melissa's struggling to hang on from the flow of the Truckee River, but she's just trying to get a hydrolab. So temperature monitoring has been going on by my shop since we started the variable in-stream flow in the early 2000s. Plus, we picked some geomorphic reaches from Tahoe Dam to Pyramid to monitor as we changed this flow regime from IFIM approach to ecosystem and variability in in-stream flow. I wanted to monitor and understand the changes in habitat along with species composition, native species composition, not just a list of species, but all of them, to see how they're responding to this natural variable in-stream flow. And so now we're going to get into, I'm just going to run through these next couple of slides because it kind of highlights the lower river is not where LCT will thrive. So this is March of 2015. It was a drought year. It was only the second year where we were managing migration. They didn't get past Derby Dam, of course, at that point. There weren't that many of them, but we're tracking the temperatures. So March, if you note the Marble Bluff, Numana Dam, up to Derby Dam, temperatures are still decent. April, temperatures are 15 to 20 degrees Celsius, still tolerable for LCT, but the temperatures are better later in the season in a drought year upstream, obviously. And then by May of that drought year, temperatures are lethal even for LCT. LCT are highly resilient in variable temperatures, but 25 degrees Celsius is their max to thrive. So the next building block, the next challenge, which is a political challenge as much as a scientific challenge, which much of this story has been, is managing the risk of hybridization. As I indicated, we're working with the Nevada Department of Wildlife. We stock LCT in lieu of them stocking rainbows to try to work with them and the community to build an understanding that when these fish migrate up from Pyramid Lake, that we want the habitat there to minimize that risk of hybridization for recovery. Cutbows have been stocked by Endow in Pyramid Lake back before the 70s, and they never survived, so Pyramid Lake is not conducive to non-native trout. But still, there are ways to manage the risk. We are just now in the data collection. It's kind of odd to demonstrate hybridization is occurring when we know trout hybridize with rainbow trout, but it is we have to build a roadmap to bring people along, fish commissions, state agencies, and communities. And so with evidence that my shop and Robert Alchokachi are collecting about how rainbow trout use the system above Derby Dam, how do the flows impact them? Because I knew at the time when the variable in-stream flow was kicked in that it would benefit the habitat, it would benefit LCT, but it would also benefit non-native trout. I knew that going in, and but now that we have LCT, we're documenting and learning how they use the system under variable in-stream flow conditions. The era of using droughts to minimize the presence and the reproductive success of rainbow trout is in the future. These river conditions before variable in-stream flow, the river was dry downtown, Reno Sparks, dry in many reaches, and so rainbow trout don't persist year to year. They do well in high water years. So managing droughts where LCT can stay in pyramid in high drought years, can damp down the presence of rainbow trout, use high water years where the habitat there's less rainbow trout present. But we have a lot of work to do before we can really get to some tangible roadmap for dealing with rainbow trout in a real meaningful way. So in summary, again, this species, Cui-ui, this habitat is remarkably resilient. As most habitats, you just understand it a little bit, tweak it a little bit, and the opportunity for just dramatic results can be realized. And I want to acknowledge, obviously the Pyramid Lake Paiute Tribe, Utah State University, USGS researchers, Mary Peacock, and I expect to see 2, 3, 4, 5,000 30-pound cutthroat trouts downtown Reno and Sparks with a trout festival as fish swim past in 5 to 8 years is my prediction.
Up to the microphone. There we go. Questions in the front or back here?
I asked my staff to jump up and clarify if they needed to clarify anything I just said. No questions?
Could you just clarify how fish got to Pilot Peak? I mean, they didn't take the train.
Well, you know,
Well, they might have.
50
Anyway, I think that that's important to point out is how'd they get out there?
Yes, you know, this, and you can speak to it as well as I can, Mary, but the state of Nevada wanted to replicate an awesome fishery that disappeared from Pyramid, and they would move them all over northern Nevada and other locations, apparently. And from the genetics, and Mary can speak to this, that the populations out in the hinterlands, the fluvial form, the northeast of Nevada, northwest of Nevada, there's no sign of Pilot Peak genetics in those populations. And the stories are varied, but they probably were taken over in a truck and moved in waters because the population was well known for its world-class fishery, and entities wanted to recreate that in small streams, and so it got translocated out in the hopes of creating a fishery out in other waters.
Howdy, great work, great talk, great fish. In the wheelhouse of cutthroat trout, there have been other similar discoveries like the greenback and the San Juan cutthroat, and they have a demonstrated extreme resilience, and they're found sometimes when they're thought extinct or to be found nowhere at all. Given these discoveries and your own miraculous discovery of the Pilot Peak genetics, is there still a continued effort searching for other isolated genetic populations?
I think that there are in other species. I will say that in my early years, I had an isolation building constructed with the anticipation there would be a unique strain of Lake Tahoe, LCT, a unique strain of Walker that we could bring in and create broodstock. I didn't realize I was water limited and space limited, but that was my vision. And then we funded Mary, and when she was able to, you missed Mary's talk probably, but great work on her part to find these museum mounts of the original strain of Tahoe and Pyramid, but also look at them in comparison to the other populations throughout the range of LCT, and found that what was thought to be original strain in the late 70s by the state of California to Lake Tahoe, Macklin Creek, did not associate with the original Tahoe museum mounts. The populations in the headwaters of the Walker are so depauperate of genetic diversity that you can't get a signal of what they're associated with. They're so inbred. And Mary, do you want to speak to any of that? But so I have one, I'm getting tired. And two, you know, we are water limited at the hatchery, and I think we've landed on the recovery population needed for the Lake Tahoe Truckee Pyramid Corridor. That's not to say that there aren't opportunities in future people or current people to investigate some of these populations out and do a similar comparison and to bring in some genetic diversity from those populations as well, if they're linked to the original. Go ahead, Mary. Sorry, I'll quit talking.
Yeah, my lab has DNA samples from probably every extant population of LCT across the range. And when we started this work, way back when, there were a lot of out-of-basin transplanted populations that people want to know what the origin of these fish were. And so we did look at a lot of different populations. None of them were this Pilot Peak strain, but we could find fish that belonged to other watersheds that were planted. But I can't say 100% that there's not another population of Pilot Peak fish out there somewhere, but I doubt it. Because, we really have, not just me, but my compatriots with Trout Unlimited and other researchers, we've scoured the landscape for this fish, and we really do have a fairly complete set of samples for the extant populations of this fish, in basin and out of basin.
But on that note, I was contacted in the 90s by a researcher in New Zealand who said that there was the original pyramid population that had been transplanted in New Zealand, and that is no exaggeration, though I can be prone to that sometimes. And the government wouldn't let me travel to New Zealand to collect samples, but New Zealand may have LCT, just a little, but that's from the 90s, so.
I have a question. Thank you for that very inspiring talk. I've worked on river restoration on the Rio Grande in New Mexico, and I know how challenging it can be.
Yeah.
My question is, if these species, if the LCT and the Cui-ui are ever delisted, would you still be able to use Stampede Lake water?
That is a very key question, and in a delisting process, that's not my area, that's ecological services, what got a species to recovery has to be maintained, but that would be a legal interpretation, and the key thing is bigger solutions than Stampede for securing water inflow to Pyramid Lake. And one of those is improving the efficiency in the Carson Division in the Newlands projects, because their diversion of the Truckee is only supposed to be supplemental to their water right on the Carson Division. And the more they can learn and develop efficiencies to live on the Carson side, the less they will need the Truckee side. But that's an excellent legal question, I would say, for future people that put forward any delisting packages.
Hey, thanks for the great talk. I had a couple questions. If you Google Pyramid Lake fishing, you see people with a bunch of huge trout holding them. Are those Pilot Peak fish?
That's correct.
Okay, cool. And then I was just hoping you could clarify where they're currently spawning in the Truckee River and where eventually you'd like to get them to. It sounds like they're just spawning below Derby Dam, but is the hope to get them to spawn all the way up to Lake Tahoe?
That is the long-term vision that I had when I was a very naive 32-year-old. But yes, I think we're about 10, 15 years away. If we can get the community, rallying behind this unique conservation, which they have, the angling community has been one of our greatest supporters. Once they could tangibly hold, the first one was caught, it was a 19-pounder, and it was a remarkable moment in the messaging for the angling community. But yes, right now, as they come up, we are holding them at Derby Dam. Let me find a map here. I can't make it. I'm trying to do my Apple, expanding it. So Derby, as you can see, no, it's not on, so you can't, you're only looking at me, great. Is it on now? So Derby is this, pardon me while I try to look, yes, is the little red dot. And that is the interbasin transfer, where they take all of this Truckee River water over to this reservoir in the Carson Basin. And right now we're keeping them below Derby, even though that habitat is, in my opinion, not sustainable habitat, but they are reproducing there. We're documenting reproduction. Reds are being formed. There was a complex of 48 reds in that right below Derby. And so right now we're just trying to grow our understanding of how this population is going to use the Truckee River. Their timing of reproduction, when do they migrate, what flows, what habitats do they use. There's a lot to learn in a little more contained environment, and as we build our support with the state of Nevada to address the risk of hybridization upstream, which we are working on presently. Because the other thing that is happening is investments. Because of the success of this conservation story, the Bureau of Reclamation invested $37 million 2 years ago for a fish screen at Derby on the canal so that we can use the fishway that was built in 2005 to pass cutthroat trout. So that $37 million investment, and then we just received payment to fund improving passage at New Mana Dam, which is an irrigation structure within the Pyramid Lake Paiute tribe, which will then, as these fish come up in number, 2,000, 4,000, 10,000, 20,000, they will be largely unimpeded once we let them pass Marble Bluff Dam. But right now we're keeping it kind of contained and a little manageable, growing, learning, adapting to our methods for sampling and tracking and monitoring with USGS.
Any other questions? Okay.
Thank you.
Thank you so much.
Thanks so much again. Just a couple quick announcements here as we head into our final afternoon of our regular program. There's a number of lunch meetings coming up here and then a full schedule of talks this afternoon. And then we do have our closing reception this evening. I just wanted to let you know that folks will be gathering kind of 10 or 15 minutes before it starts in the registration area if you want to walk over with the group. And we also did add a few more tickets. I've heard a couple people asking, and so if you didn't get one originally and you're interested in joining us this evening for some bocce ball and some drinks and some more conversation, please go ahead and snag one of those, and we'll look forward to seeing you this afternoon and this evening. Thank you.